All right. So one thing about that often gets asked about on AP exams, especially in the FRQ part of the test, is that you may be asked to describe an experiment, and you've got to know the things that have to be in that experimental design for it to work. A hypothesis is really, really important. When you're writing a hypothesis for AP Bio, I want you to write it in the form of an if-then statement, and you're going to write if. You're going to describe what should happen to the independent variable, the variable that you're intentionally manipulating. And then you're going to write then and tell what's going to happen to the dependent variable, the variable that's responding because of changes to the independent variable. For example, if you were working for a fertilizer company like the Miracle-Gro company, you might say if Miracle-Gro is applied to tomato plants, so the Miracle-Gro would be the independent variable, then they will grow taller. So their height would be the dependent variable. So if then. Now, some other really important things to talk about when you're doing experimental design is that you're always going to divide your subjects in your experiment into at least two groups, an experimental group and a control group. The experimental group is the group that you're going to expose to the independent variable. For example, since all this COVID stuff is going on, let's use that as an example. Let's say that you're testing some kind of treatment for COVID. The people in the experimental group are going to get that treatment. That's the independent variable. The people in the control group are not going to get the treatment. And that might sound cruel for you not to give them the treatment, but you've got to know if the treatment works. The dependent variable in that kind of experiment might be, do their symptoms go away? It might be, do they live or die? You've got to decide what your dependent variable is. If we're talking about our Miracle-Gro experiment, the experimental group would be the plants that are actually given the Miracle-Gro. Now, you might have plants that get different concentrations of Miracle-Gro, and that might happen in a drug study too. Some might get 10 milliliters, some might get 20, some might get 30, and that's okay. But still the independent variable is Miracle-Gro. The amount may vary between subgroups. The control group is typically not going to get any. It's the no treatment group, so that you can tell what the actual treatment is doing. The most common type of control group is a negative control group. And again, that's a control group that is not exposed to the experimental treatment. So in a drug study, those individuals in the negative control group don't get the drug. Another example is, let's say you wanted to know if there's bacteria growing on lettuce. So you might set up an experiment where you wipe a lettuce leaf with a swab, and then you wipe that swab over an agar plate to see if it grows. A negative control group might be just to take a sterile swab and wipe it on the plate. You wouldn't expect anything to grow. If something does grow, that tells you there's a problem with your setup. Maybe your swabs are contaminated or your plates are contaminated. Now, there are some cases where you might also want a positive control group. This is a group that's not exposed to the experimental treatment, but it is exposed to some other treatment, a treatment that you know has an effect. For example, let's go back to the swab experiment in the lettuce. So as a positive control, you might take that swab and wipe it on a plate of actual bacteria, and then wipe it on the plates that you're using to grow your bacteria in your experiment. You would expect growth to be there. If you don't get growth, that tells you there's either something going on with your swabs, or maybe something going on with your plates. Maybe the bacteria can't grow on the plates. Maybe there's an antibiotic or some kind of impurity in the plates. Or maybe the temperature you're trying to grow those bacteria in is too high. There are cases where an experiment may have both negative and positive control groups, but in most cases in AP Bio, you're going to see negative control groups. Groups that where the subjects in those groups are not exposed to the independent variable. That's the most common thing to see in AP Bio. I hear some background noise from someone's mic, So please make sure your mics are muted. Thank you, guys. So another thing to think about in experimental design are constants. And these are factors that you need to keep the same amongst all groups, experimental and control. You really want the only difference between your control group and your experimental groups to be that one independent variable. Everything else should be the same. For example, in the Miracle-Gro study, you would want to be using the same kinds of plants, growing in the same kind of soil, the same amount of light, the same amount of water, the same amount of nutrients other than Miracle-Gro in the soil. Everything should be the same except Miracle-Gro or no Miracle-Gro, or maybe the amount of Miracle-Gro given. That could change. But these things are called constants, things that should be the same between all members of all groups in your study. And this is just to make sure that the changes that happen are due to the independent variable, not some other outside force. Some other things to think about in experimental design are sample size. If you're asked to design an experiment in an AP Bio FRQ, you need to make sure that you describe to the people reading your answer that you're going to have a large sample size. I wouldn't try to say a certain number of individuals. I would just say you've got to have a lot of individuals. You can't have one plant in one group and one in another. You need a lot of plants in your control group and a lot of plants in your experimental group. You also need to do randomization. And what that means is which individuals are assigned to the control group and which are assigned to the experimental group. That's determined randomly. That's the cut down on bias. If you were someone conducting a drug study, you might unconsciously try to essentially rig the study by putting certain people into each group. You don't want that to happen, so you let a computer essentially randomly assign individuals to each group, and that cuts down on the bias. Another important thing to think about is replication. Study can't just be done once. You need to run multiple trials, and also you need other individuals to run the same experiment. Science is always about replication, doing that experiment more than one time. So that's all we're going to say about experimental design for right now. That's going to show up on the AP exam. Later, as we do more of these sessions, we're going to look at some free response questions, and we'll talk a lot more about experimental design there. But what I really want to focus on today is data analysis and statistics. Those are things that a lot of you probably aren't very familiar with, and I want to make sure you leave this session being somewhat familiar with them. So when you're analyzing data, you're trying to arrive at conclusions about the data. You want to be able to use your data to make some claims. You want to be able to support some arguments you might have using your data. You also want to get an idea of how reliable your data are. And you want to be able to communicate your conclusions to other people in the scientific community. So scientists do data analysis in most cases by using statistics. We're going to look at some very simple statistics today. We could get a lot deeper than this, but we're not. We're going to talk about two categories of statistics, descriptive and inferential. So descriptive statistics basically describe your data. These are things like measures of central tendency, types of stats that talk about what's the middle number in your data set. We're talking here about things like the mean, which in AP Bio, you're going to see symbolized with an X with a bar on top. It's just the average. The median, the middle number in a set, and the mode, the most common number in your data set. We're also going to hear about standard deviation, which we'll talk more about in a minute. And we also might talk about the standard error of the mean. Those are going to be the most common types of descriptive statistics that we'll use. And again, they're just used to describe your data, to let you get a feel for what your data are like. Inferential statistics, on the other hand, are used to make predictions from a sample about a population. And that word sample is really important. So let's say we were studying a group of plants in a field. And let's just go back to the Miracle-Gro experiment. Let's say we put Miracle-Gro on the plants in that field, and we want to know the effect that the Miracle-Gro had on the height of the plants. In most cases, we're not going to be able to measure the height of every plant in that group. The whole group of plants is our true population. Instead, we're going to take a random sample of plants and we're going to measure those and see what happens to their changes in height. Those plants that we take at random, those are a sample. And if we take them randomly and we get a big enough sample, we're hoping that the sample numbers actually reflect what's going on with the entire population. That's the basis of inferential statistics, using a random sample to predict things about the entire population. So again, there's some descriptive statistics we already talked about: the mean, the standard deviation, and the standard error. I'll mention sample size here. We're going to see mean abbreviated with an X bar. The sample size, the how many individuals are in your sample, you're going to see that notated with N, just a number N, for number in sample. Standard deviation, we're going to see abbreviated different ways. Sometimes it's S, sometimes it's SD. Most of the time it's going to be S. Standard error of the mean is going to be SEM or SE, and then like X bar, like so. Now this formula right here looks really, really confusing. It is on the AP Bio formula sheet. This is the formula for the mean, the average. I wouldn't even worry about knowing that formula. All of you know how to calculate the average. That means to take each individual data point, that XI is the individual data points. This means to sum them up. It's the summation symbol, so add up all the data points. And then this means divide by the sample size. So divide by the number of individual data points. You guys all know how to find the mean. Don't let this confusing equation be a problem. Now, standard deviation is a little more tricky. This is the formula for standard deviation. Now, I will tell you, this formula is also on your AP Bio formula sheet, and it actually has a little asterisk beside it on the formula sheet. And if you look down to see what that asterisk means, it tells you that you're not actually going to have to calculate a standard deviation on the AP Bio exam, but you're going to be asked to interpret what it means and also maybe use it to do some graphs and make some conclusions. I want you to be able to calculate it though, so you understand what it is. Essentially, the standard deviation tells us how far our data points are from the mean of the data set. When you hear standard deviation, I want the word variation or variance to come to your mind. It's essentially telling you how different your data points are from each other and from the mean of the data set. It's about variation. Higher standard deviation means more variation. Okay. Standard deviation is about variation. And again, we'll talk more about this formula in a second when we work an example, but this XI is each individual data point. The X bar is the mean of the data set, and N is the sample size. So we'll actually show you how to calculate a standard deviation on an example problem here in just a second. So this is showing you what, if you graphed your data sets, what it might look like. And essentially you're graphing how many data points are at each value. Numbers with a low standard deviation are going to be very closely clustered together and clustered around the mean. This is the mean right here. On the other hand, data sets with a lot of standard deviation, a high standard deviation, are going to be spread out. And there's going to be data points that are very far from the mean. Yes. All right, so again, values with a low standard deviation clustered around the mean. Values with their data sets with a high standard deviation spread out. Lots of the points may be far from the mean. Here's another way to look at that with a line graph. Here we see data points kind of all over the place around the mean. The mean is the line. Over here with a low standard deviation, the mean is again the line, and all the data points are clustered closely around it. Low standard deviation versus high standard deviation. If you're doing an experiment and you're wanting to make some conclusions, you're going to be a lot more confident in this data, the data with a low standard deviation, than you are in the data with a high standard deviation. We might say that this data is more reliable. It's also more precise. The standard error of the mean, on the other hand, remember, is used to estimate a population mean, a true population mean, from a sample mean. So we're taking a small sample of individuals, and we're going to use that sample to project out what we think the true population mean is. Here's the formula. This one is also on your AP Bio formula sheet. So standard error of the mean is equal to the standard deviation of the sample divided by the square root of the sample size. There's that n again. And again, we're going to see example problems here in just 1 second. Another thing that often happens in graphs is we're going to use these things called error bars, and we're going to use them to create what's called a 95% confidence interval. And instead of trying to read through this whole slide with you, I'm going to talk about this as we do an example problem, and we'll talk about what it means also as we go through the example problem. Let's go ahead and just look at this example problem. So each of 3 groups of young rabbits are fed a different diet. All other factors in the rabbits' environments were identical. So these are the constants. So we might say they're housed in the same kind of housing. They're given the same amount of water. They're kept at the same temperature. They're the same type of rabbit. Everything's the same except their diet. So we're measuring the change in mass of each rabbit over a 4-week period. That change in mass is our dependent variable. Our independent variable is the type of diet. So we're intentionally varying the diet, independent variable, and we're measuring what effect that has on the mass of the rabbits. So now it's asking us to statistically analyze the results by answering the questions listed below about the data sets A, B, C. So we have 3 groups of rabbits. We have rabbits that are grass-fed, rabbits that are fed brand A food, and rabbits that are fed brand B food. And we're looking here at their changes in mass over that 4-week period. And the first thing that we're asked to do is calculate the mean of each data set. Well, you guys know, again, to calculate a mean, you just add up the values. So for the first set, it would be 4 plus 4 plus 5 plus 8 plus 10 plus 11. Add those up. And in this case, our sample size is 6. So we would add them up and divide by 6. And we're going to do that for each group. I've already done that work on the next slide. So here we see the means here. So we've calculated the mean of set A to be 7 grams, the mean of set B to be 11 grams, the mean of set C to be 5 grams. Now the next thing asks us to calculate the standard deviation. Now remember, that's a measure of variation, variance. Higher standard deviations mean the data are more spread out, they're more different from each other and different from the mean. So we're going to look first at, this is just data set A. These were our individual values for set A, 4, 4, 5, 8, 10, 11. It's easier to calculate standard deviation if you don't have a graphing calculator using a table. So what I've done first is I've taken this part of the equation from under the radical, xi minus x bar. So I took my first individual data point, 4, and subtracted from it the mean, which was 7. 4 minus 7 to get -3. Here, I've got a mistake in my calculation here. So this one should also be 4 minus 7 to get negative 3. And this ought to be 9. The next one should be 5 minus 7 to get negative 2. 8 minus 7 to get 1. 10 minus 7 to get 3. 11 minus 7 to get 4. So in my next column, I take this column and essentially square it, because that's what's under my radical there, xi minus x bar squared. So negative 3 squared, remember, negative times a negative is a positive, is 9. Negative 3 squared is 9. Negative 2 squared is 4. 1 squared is 1. 3 squared is 9. 4 squared is 16. Now, the next thing tells me to sum up xi minus x bar squared. So I need to add all those up. So 9 plus 9 is 18. Plus 4, 22. Plus 1, 23. Plus 9, 32. Plus 16 is 48. So the summation of xi minus x bar squared is 48. Now I want to divide that by n minus 1. Remember that n is your sample size. Our sample size is 6. We had 6 values. Don't forget to turn off your mic, guys. So we're going to go 48 divided by 5 in this case. And then we need to take the square root of that whole quantity. The 48 divided by 5 is 9.6. We're going to take the square root of that whole thing, and we end up with a standard deviation. Guys, please turn off your mics, of 3.098. And again, I had a mess up there on my slide. So this is the correct standard deviation. 3.098. Now we're going to also calculate the standard error of the mean using this formula. So the standard error of the mean is the standard deviation, in this case 3.098, divided by the square root of the sample size. Sample size is 6. So 3.098 divided by the square root of 6. Guys, somebody still has on your mic. Please turn that off. So we end up with a standard error of 1.265. All right, I'm going to write down these values just so that I have them on the next slide. So for, again, for data set A, our standard deviation is 3.098. And our standard error of the mean is 1.265. And we also, for that group, we have a mean of 7 grams. All right, now we're going to go to set B and do the same kind of thing. So here's set B. Again, we're going to take each of our individual data points, 8, 8, 9, 11, 14, and 16. And we're first going to subtract the mean from each of those. So the mean for set B was 11. We're going to go 8 minus 11 to get negative 3. Again, 8 minus 11 to get negative 3. 9 minus 11 to get negative 2. 11 minus 11 to get 0. 14 minus 11 to get 3. And 16 minus 11 to get 5. Where they're going to square each one of those values. So negative 3 squared to get 9, negative 3 squared to get 9, negative 2 squared to get 4, 0, 3 squared to get 9, and 5 squared to get 25. We next need to sum up those data points. So 9 plus 9 is 18, plus 4 is 22, plus 9 is 31, plus 25 is 56. So we get 56 over n minus 1. Well, again, the sample size is 6. So sample size minus 1 is 5. And we're going to take the square root of that whole quantity. So 56 divided by 5. Take the square root of that whole thing. to get a standard deviation of 3.47. We're then going to go back to this equation right here, and we're going to calculate the standard error of the mean. So we're going to take our standard deviation of 3.347, and we're going to divide that by the square root of our sample size, which was 6. You do that calculation, you end up with a standard error of the mean of 1.367. I'm going to repeat the calculation for set C, and then we'll look at what we really need to know how to do, which is how to use these numbers to make graphs and interpret those graphs. So bear with me for just a minute as we calculate data set C. Here's data set C. So 2, 4, 4, 5, 6, and 9 were our individual data points. Our mean for this set was 5. So we're going to go 2 minus 5 to get -3, 4 minus 5 to get -1, 4 minus 5 to get -1, 5 minus 5 to get 0, 6 minus 5 to get 1, and 9 minus 5 to get 4. We're going to square each of those values. See that happening here. And then we're going to sum up this entire column. So 9 plus 1 is 10, plus 1 is 11, plus 1 is 12, plus 16 is 28. So we end up, this part of our equation up here is 28. We're going to divide that by n minus 1. Again, sample size is 6, so n minus 1 is 5. We're going to take the square root of that whole quantity. So 28 divided by 5. Take the square root of the whole thing. We end up with 2.366 is our standard deviation. We'll then use this equation to get our standard error of the mean. So we're going to take our standard deviation, 2.366. We're going to divide that by the square root of our sample size, which is 6, to end up with a standard error of the mean of 0.966. And again, I'm going to make sure I have all these data points written down so that we have them for our graph, which we're going to do in just a second. And that's really the important thing, is the graph. So for set C, we have a mean of 5, a standard deviation of 2.366, and a standard error of the mean of 0.966. I didn't write down the values for set B, so I'm going to go back and do that as well. So for set B, our mean is 11. Our standard deviation is 3.347. And our standard error of the mean is 1.367. So let me get to another slide. I'm just going to write on this graph for a second. So for set A, we had a mean of 7. We had a standard deviation of 3.098. And we had a standard error of the mean of 1.265. For B, we had a mean of 11. We had a standard deviation of 3.347. And we had a standard error of the mean of 1.367. And for C, we had a mean of 5. We had a standard deviation of 2.366. And we had a standard error of the mean of 0.966. So one thing I want us to look at are these standard deviations. Here's the one for A. Here's the one for B. Here's the one for C. Notice C's standard deviation is quite a bit lower than that of B and A. And what that tells you is that the data points in set C were closer together. They were clustered more closely to each other and to the mean of that whole data set. Whereas the values in A and B were a little bit more variable. There was more variation in the measurements in sets A and B. So let's go back and see what else this question asks us to do. So notice that we've done A, we've done B. We talked about the standard deviation. The group with the largest standard deviation was group B. And again, that tells us that those data points are further from each other and further from the mean of set B's data set. We talked about what the standard deviations tell us. They tell us about the variation, how much the data points vary from each other and from the mean of the data set. We calculated the standard errors of the mean for each data set. Now, the important thing that you really need to leave this session knowing how to do are parts F and part G. All right, we had some minor technical issues there, so let's try to go ahead and create the error bars and the graphs of the means. And we'll talk about what 95% confidence intervals and error bars and all that stuff means as we go. First of all, though, we want to graph the mean change in mass, and we always want to graph our dependent variable on our y-axis. So in this case, the mean change in mass, that's going to be our dependent variable. We measure that in grams. We always put our units in parentheses. Our independent variable, on the other hand, goes on our x-axis, and it's the group or the type of food, the diet. So there's not really a lot to put there in terms of units. So we're just going to label that as diet. Really, on the y-axis, all we have to graph are our groups. So we've got group A, and I'm just going to evenly space those about 3 lines apart. We've got group B, and then we've got group C. First of all, we just want to draw bar graphs or histograms for the means. So we need to label our axes in terms of numbers, our y-axis at least. And I think with the graph that I got, 2 is going to be the appropriate increment size. So group A had a mean of 7. So let's go up to 7, somewhere about right here. And let's draw down a bar graph. Group B had a mean of 11. So let's go up to about where 11 would be, somewhere in this neighborhood. And let's draw a bar graph. Again, these are not going to be perfect graphs because I'm doing them by hand here on the computer screen. And finally, group C had a mean of 5. That would put us right in this neighborhood. Something like so. All right, so to do 95% confidence intervals, you're going to take your mean and your standard error of the mean. And you're going to go up from that mean 2 times the standard error of the mean. So we're going to go 2 times 1.265. That gives us 2.53. We're going to add that to our mean. So we're going to go 7 plus 2 times the standard error of the mean. And that's going to put us at 9.53. So we're going to go up to 9.53 for group A, and we're going to put a little error bar, a little horizontal line, and we're going to connect that with a vertical line down to the mean. You don't have to label the numerical value of that, but I like to, 9.53. We also need to subtract 2 times the standard error of the mean from the mean. So we're going to take our mean, 7, minus 2 times that standard error, so that's minus 2.53. That puts us at 4.47. And again, we draw another little horizontal line. We connect that up to the mean like so. So this is what we call a 95% confidence interval. It's up and down from the mean, 2 standard errors of the mean. And it's called a 95% confidence interval because we can be 95% sure that the true population mean lies somewhere between 9.53 and 4.47. Our sample mean was 7, but that was just for a very small group of organisms. Our true population mean we're projecting or inferring to be somewhere between this upper bound of 9.53 and the lower bound of 4.47. So let's do the same calculation for B. Let's take the standard error of the mean and multiply it by 2. So that's 1.367 times 2. That's 2.734. We need to go up from the mean 2.734. So 11 plus 2.734 is going to put us at 13.734, somewhere right in there. Draw down a vertical line, connect to the mean. We also need to subtract 2 times the standard error from the mean. So 11 minus 2.734. That gives us 8.266, which will be somewhere in this neighborhood. So we can be 95% sure for diet B that the true population mean lies somewhere between 13.734 and 8.260. Same calculation for part C. Take the standard error of the mean and multiply it by 2 to get 1.932. Add that to the mean, so 5 plus 1.932 puts us at 6.932, somewhere right in here. Connect that to the mean with a vertical bar. Then we're going to subtract 2 times the standard error from the mean, so 5 minus 1.932 to get 3.068. So that's going to put us somewhere out in here. So we can be 95% sure that the true population mean for the rabbits given diet C are between 6.932 and 3.068. Now the question asks us, based on these sample means and SEMs, are the mean changes in mass amongst the 3 groups statistically significant? And justify your answer. What you need to look for when you're looking at error bars is do the confidence intervals overlap? So notice that between groups A and B, the error bars overlap right in this area right here. What that means is even though their sample means are fairly different, the true population means could both lie in this same area. What that tells you, when error bars overlap, the differences between the treatment groups are not significant. So overlap between error bars or confidence intervals means that the differences are not significant. Notice though, if we compare B and C, there's no overlap between the confidence interval for B and C. When there's no overlap, that leads you to believe that the differences between the groups are significant. So essentially what that's telling us here is that diet B seemed to have caused rabbits to gain more mass than diet C did, and those differences were significant. Notice if we compare A and C, those groups overlap right in this area. And because they overlap, that tells us that the differences between diet A and diet C are not significant. So when there's overlap between error bars, the differences between the groups are not significant. And when there's no overlap, the differences are likely to be significant. That's all you need to say to justify your answer on these problems. Got one other quick question I want us to look at, and that's this. So here we have some adult male guppies fish, and they have genetically determined spots. Well, we grow these fish in different areas, some with a predator and some without, and we measure how the mean number of spots changes across time. What I really want you to pay attention to here is only part A, because it's the part that deals with the stats that we're looking at now. Describe the change in genetic variation in the population between 0 and 6 months, and provide reasoning for your description based on the means and SEMs. So we're only looking at time 0 right here and time 6 months right there. And what I hope you notice is that the confidence interval, the error bars here are really, really tall, whereas here they're really small. Well, remember that those error bars are based on SEM, and that SEM comes from standard deviation. And that standard deviation is really a measure of variation. So taller error bars mean higher SEM, which means a greater standard deviation, which means more genetic variation. So the answer to this question would be, at time 0, there's a lot of genetic variation because of the tall error bars, the high SEM. But as the population moves towards 6 months of age, that genetic variation greatly decreases. And we know that again because the size of the error bars, and therefore the size of the SEM at 6 months, is much, much smaller. All right, that's it for our session today. I hope that helps, and we'll do this the same time next week.